Hello everyone, and welcome to the first webinar in our 2024 Cannabinoid Conundrums webinar series. Today we will hear from Dr. Lugo Song, Associate Professor in the Department of Chemistry at Western Illinois University, on LC-DAD with optional ESI MS, an emerging gold standard for hemp compliance testing. Without further delay, I will turn things over to Dr. Song.
Thank you, Kelsey, for your kind introduction. Hello, everybody. Welcome. My name is Lugo Song, and I am from Western Illinois University. Here we have forensic chemistry program, and I am the program director. Here's my contacting information, and this is the title of today's presentation. So first, some introduction here. In the USA, the Controlled Substance Act of 1970 classified all parts of the plant cannabis sativa oil as a Schedule I controlled substance. Therefore, the statute retained the control over all botanic varieties of the plant, including marijuana and hemp. Now, the 2018 Farm Bill legalized hemp as the plant with a delta-9 THC concentration of not more than 0.3% on a dry weight basis. Cannabis sativa oil is well known for its characteristics to produce cannabinoids. The plant initially bisynthesized CBGA, which is then bisynthesized into CBDA, delta-9 THCA, and CBCA. Subsequent non-enzymatic decapsulation of the acidic cannabinoids generate neutral cannabinoids. CBG from CBGA, CBD from CBDA, delta-9 THC from delta-9 THCA, and CBC from CBCA. Other cannabinoids, including CBNA, CBLA, CBN, CBL, delta-8 THC, and CBT, are the results of non-enzymatic isomerization, decapsulation, decomposition, and/or degradation. The plant also produces cannabinoids which differ in the length of the side alkyl chain. 5 carbons is the most abundant. 3 carbons is the second most abundant. For example, CBDVA corresponding to CBDA, THCVA corresponding to THCA, CBDV corresponding to CBD, and THCV corresponding to THC with 5 carbons in the side chain. So far, the maximum number of cannabinoids that have been quantified in hemp plant material are 18, as shown over here with the structures. In our study, ACBD was used as the internal standard because ACBD or abnormal CBD is not naturally present in the cannabis plant. Our study also used the 6 stable isotopically labeled cannabinoids, as shown over here, plus ACBD D3 in LC-ESI MS analysis. We didn't use 18 isotopically labeled cannabinoids because some of them were not available and some of them were available but very expensive. Now, an introduction to the hemp and hemp-derived products. Non-psychotropic cannabinoids-based products are derived from legal hemp, including hemp plant materials, concentrates, whipping oil, tinctures, edibles, topicals, and PET sheets. Psychotropic cannabinoids were produced by legal argument that the 2018 Farm Bill legalized the sale of them due to their natural presence in hemp, despite in amounts too small to produce psychotropic effects, and or because they are derived from CBD. Those include the double bond position isomers of delta-9 THC, for example, delta-8, delta-9, 11, delta-10, there are two isomers here, and delta-6, 8, 10, THC, homologs of delta-9 THC and delta-8 THC, including the side chain with 8 carbons, 7 carbons, 6 carbons, and 4 carbons. Analogs of delta-9, delta-8 THC, including the O-acetate, hexahydrocannabinoids, or HHC, and HHC-O acetate. Now, psychotropic cannabinoids-based products include distillates, vaping cartridges, tinctures, and edibles, as well as fortified hemp plant materials and fortified non-psychotropic cannabinoids-based products listed over here. Hemp compliance testing often refers to the analysis of plant materials. It is subject to regulation by the USDA. Bottom line, hemp should not have more than 0.3% total content of THC derived from the sum of delta-9 THC and the precursor delta-9 THCA. Now, hemp compliance testing can often refer to the analysis of hemp-derived products. The 2018 Farm Bill by the Congress and the 2021 final rule by the USDA require that total content of THC is not more than 0.3%. And additionally, there are other regulations to follow. Potency testing is aimed to quantify all the major cannabinoids so that the quality and safety of hemp and hemp-derived products intended for human consumption can be determined. Since 2017, the AOAC International have developed standard method performance requirements for potency testing of cannabinoids in cannabis concentrate, dried plant materials, hemp, edible chocolate, and beverages. Those numbers has a range, and they are the content range for each individual sample. 5 cannabinoids are in the required list: CBD, CBDA, CBN, delta-9 THC, and delta-9 THCA. 9 cannabinoids are in the desired list: CBG, CBGA, CBC, CBCA, CBDV, CBDVA, delta-8 THC, THCV, and THCVA. We covered previously. In our study, we consider additional 4 cannabinoids because CBNA and CBT were often found in the samples. However, CBL and CBLA were occasionally found in the samples. So far, the maximum number of cannabinoids that have been quantified is 18. Food contamination testing is meant to detect cannabinoids as contaminants in food containing hemp seed and hemp seed-derived ingredient. It is required in Canada and in EU. In the USA, however, the DEA used the 0.3% total contents of THC to exempt hemp seed and hemp seed-derived ingredients from controlled substances. Well, the FDA concluded these products were generally regarded as safe. In other words, we usually don't do food contamination testing in this country. GCFID and GCMS methods have a long-standing history for cannabinoids testing, but acidic cannabinoids can be thermally decarboxylated to their neutral counterparts at the injection port. LC-MS methods are suitable for not only hemp compliance and potency, but also food contamination testing. As we all know, it has a superior sensitivity and selectivity. However, the methods demand expensive instruments that are usually unavailable and inappropriate for routine analysis by crime labs. LCDAD methods have emerged as the best approach in hemp compliance and potency testing because the required LQ is relatively higher and the instrumentation is widely accessible by crime labs. However, additional MS confirmation of identity is necessary according to the SWG drug recommendations as follows. So far, all published LCDAD methods have been developed for potency testing. From an analytic point of view, this method can be readily adopted in hemp compliance testing. Methods that have been developed specifically for hemp compliance testing can be hardly found. A method developed specifically for hemp compliance testing can be simpler and faster because quantification of cannabinoids other than delta-9 THC and delta-9 THCA is not required. On the other hand, a method developed specifically for hemp compliance testing must demonstrate that quantification of delta-9 THC and delta-9 THCA is not interfered by any other cannabinoids in the sample. Now, the objectives of this presentation to elucidate the key factors to use LCDAD with optional ESI-MS for compliance testing of hemp and also hemp-derived products through a summary of 4 published methods and 2 methods under peer review for publication by our research group. To discuss the essential aspects for best practice to implement LCDAD with optional ESI-MS for compliance testing of hemp and hemp-derived products. To seek for collaboration with crime labs for research on a best practice to implement LCDAD with optional ESI-MS for compliance testing of hemp and also hemp-derived products. Here are the contacting information. Now we proceed to the experimental. Chemicals, standards, calibration solutions, and QC samples are listed here. It is noted that the PDF version of the slides will be available as an archived resource. So I'm not going through the details of these. Samples that have been analyzed included hemp concentrates, plant materials of marijuana, marijuana cigarettes, hemp flowers, hemp cigarettes, delta-8 THC fortified hemp flowers, more hemp flowers, more delta-8 THC fortified hemp flowers, tablets, drinks, water-soluble oils, topicals, gummies, hard candy and coffee, snacks, and pet treats. Now, for easy to follow, the outline of sample preparation is introduced over here. Samples were divided into 3 categories, and the sample preparation included 6 steps for every sample category. The sample preparation of category 1 samples, homogenization. Solid samples, including plant materials, tablets, were ground using a lab blender and then pulverized using a homogenizer first. For liquid samples, including drinks, water-soluble oils, and terpene-rich topical serum, they were ultrasonicated, the whole package. Lip balms was melted inside a 60-degree centigrade water bath and vortexed for a couple of times. Body lotion and face cream were polarized using a homogenizer listed here. Ultrasonication for 20 minutes after appropriate amount of the sample and appropriate volume of methanol and appropriate amount of abnormal CBD is spiked into the mixture. The same for all the samples. Centrifugation at 13,000 RPM for 10 minutes. Filtration with a 0.2 micrometer PTFE syringe filter. Dilution with methanol for LCDAD ESI top MS analysis. Now the final concentration was determined by the type of sample. Concentrates, 20 microgram per milliliter. Plant materials, either 50 or 100 microgram per milliliter. Drinks, 2.5 milligram per milliliter. Or other samples, 250 microgram per milliliter. For LCESI MS analysis, samples were further mixed with a mixture solution in methanol containing 6 stable isotopic labeled cannabinoids and also ACBDD3 at 1 microgram per milliliter in a 1:1 volume ratio. However, we do not usually recommend this analysis. Now, sample preparation of the category 2 samples, they are sugar-rich samples, such as gummies, hard candy, and coffee. For homogenization, 10 to 20 pieces were first chopped using a lab blender, some details over here. Then 1 gram sample and 4 milliliter water were pulverized using a homogenizer. Ultrasonication for 20 minutes after 1.8 milliliter methanol, 2 milliliter 75 microgram per milliliter ACBD methanol, and 200 microliter 250 milligram per milliliter sample prepared in step 1 are mixed together. Centrifugation, filtration, and dilution were the same as described previously. 200 microgram per milliliter sample were analyzed again. We use this concentration a lot. Sample preparation of category 3 samples, including snacks and pet treats. For homogenization, samples were chopped using a lab blender. For pulverization and ultrasonication, 1 gram sample and 4 milliliter, 0.25 milligram per milliliter ACBD in methanol were pulverized by using a homogenizer, which was followed by ultrasonication, and this process is repeated. Now, centrifugation, filtration, dilution they were the same as previously described. Again, 250 microgram per milliliter sample was analyzed. Now, LCDAD, we used an Agilent 1260 Infinity 2 LC system. Those we are going to talk about later. And the ESI TOF-MS and the ESI MS-MS use an Agilent 6545 Q-TOF mass spectrometer with an Agilent jet spray ESI source operating in positive ion mode. Some MS parameters are here, and additional ESI MS-MS parameters in this table, including precursor ion, collision energy, quantifier ion, qualifier ion, and we used 6 stable isotopically labeled cannabinoids plus a CBD-D3 as internal standard. Here are the results. So separation used for compliance testing of plant materials by LCDAD ESI TOF-MS is demonstrated in this chromatogram. It is noted that for compliance testing, the baseline separation of delta-9 THC and delta-9 THCA from other cannabinoids was essential. However, the coelution of other cannabinoids didn't matter. Therefore, we were able to use our rapid separation here. Now, separation used for potency testing of plant materials by LCDAD ESI TOF-MS is demonstrated with this chromatogram. Again, for potency testing, the baseline separation of every individual cannabinoids from each other was essential. Separation with an alternative selectivity used for potency testing of plant materials by LCDAD ESI TOF-MS is demonstrated in this chromatogram. Now, here, the mobile phase changed. So wherever the mobile phase changed, you are going to expect the baseline shift. This will affect the quantification of cannabinoids on drifted baseline. However, this problem can be solved if you can detect those cannabinoids with UV detection at a longer wavelength than 230 nanometers. Now, separation used for potency testing of hemp concentrates and hemp-derived products by LCDAD ESI TOF-MS is demonstrated with this chromatogram. Again, baseline separation of every individual cannabinoids from each other was essential. However, there was a correlation here, correlation of CBG and THCV. It was noted that for concentrate and hemp-derived products, acidic cannabinoids were hardly found, especially for low abundance acidic cannabinoids like THCVA. This one was hardly found even in hemp plant materials. Separation used for potency testing of hemp-derived products by LC-ESI MS-MS demonstrated with this chromatogram. So you can see the correlation CBG, THCVA, and CBLA, CBL. However, They are not structural isomers. They have different precursor ions. However, for potency testing, again, we need the baseline separation of all the structural isomers. They have the same precursor ions. That's including, for example, CBD, ACBD, delta 9 THC, delta 8 THC, CBL, CBC, and CBT. LC chromatograms were acquired as 230, 251, 261, 269, and 285 nanometers. Most neutral cannabinoids, they have one apex lower than 230 nanometers. So they were quantified at 230 nanometer to be consistent with published results. It's noted that because they cannot be detected with longer wavelengths, so if the peaks were on a drifting baseline, you are going to have trouble for quantification. Other cannabinoids were quantified at 230 nanometer for linear calibration range between 0.02 to 25 microgram per milliliter and better sensitivity. But at their second apex 269 to 85 nanometer for linear calibration range between 0.04 to 50 microgram per milliliter and better selectivity. And also with a longer UV detection, you can quantify peaks on a drifting baseline. CBCA and CBNA over here have one apex, so we have no choice. They were quantified at 251 and 261 nanometer. Again, because of longer wavelengths, you can quantify them on a drifting baseline. External standard calibration was chosen over internal standard calibration due to simple procedure and good performance. The linear calibration range of all cannabinoids in methanol were between 0.02 to 25 microgram per milliliter. For plant materials, method 1 and 0.04 to 50 microgram per milliliter for plant materials, method 2. So those two methods were described in the sample preparation. Plotting peak area of each cannabinoids versus its concentration after a weighing factor of a reciprocal square of concentration was applied. The linear calibration range for different samples are as follows: sample type, concentration of the sample, and the calibration range converted to percentage. You can see the calibration range for every type of sample met the requirement by the AOC International, listed here. The smallest R-square of all the calibration curves was 0.9880. Now, 0.02 microgram per milliliter LLQ for linear calibration range between 0.02 to 25 microgram per milliliter is demonstrated in this chromatogram. Here, 0.04 microgram per milliliter LLQ for linear calibration range between 0.04 to 50 microgram per milliliter is demonstrated in this chromatogram. Again, if you have a mobile phase change, you are going to expect drifted baseline. Then the quantification can only be done with cannabinoids that can be detected as longer wavelengths other than 230 nanometer. Accuracy met the ISO 17025 guidelines. Precision also met the same guideline. and ACBD, abnormal CBD, a cannabinoid not naturally present in cannabis plants and commercially available with a reasonable price, was spiked into each sample to track recovery of sample preparation in real time. So those are the sample types, and those are the spiked level. We try to use 0.3%. However, drinks have a lower concentration of cannabinoids, so we spike the ACBD at a lower level. Snacks and pet treat, the sample preparation procedure required a large amount of the ACBD. So again, we spiked at a lower level to save some money. Recovery, precision, those are listed over here. They are in triplicate. ESI top MS was used to examine selectivity, indicating overall good selectivity with only a few minor interferences from the matrix at close to LLQ level. A baseline separation of the cannabinoids, especially for CBD, CBG, and delta-9.8-THC critical pairs, was essential. Due to the high hydrophobic nature of cannabinoids, many less hydrophobic cannabinoids in hemp-infused edibles would not result in interfering matrix peaks in the LC-UV chromatograms. Well, some of hydrophobic lipids and emulsifiers in hemp-derived products might correlate with cannabinoids. They were unlikely to result in intense matrix peaks due to lack of UV absorption. Relative retention time of individual cannabinoids to the internal standard ACBD was effective in avoiding false positive identification from matrix peaks. LLQs also served as an appropriate cutoff to avoid false positive identification from matrix peaks. So it was very important to choose a good combination of a percentage LLQ and a sample concentration based on the concentration LLQ of 0.02 or 0.04 microgram per milliliter, so that matrix peaks would not become intense enough to cause false positive identifications. UV detection at multiple wavelengths discovered the correlation of cannabinoids with different UV absorption spectrum. For example, CBDVA versus CBNA isomer, CBNA versus a delta-9 THC isomer, and CBC versus a CBG isomer. However, UV detection at multiple wavelengths could not discover correlation of cannabinoids with similar UV absorption spectrum, for example, CBDV versus a delta-9 THC isomer. Nevertheless, no interference in the accurate quantification of delta-9 THC and delta-9 THCA were observed. This chromatogram demonstrates the interference to quantification at close to LLQ level. You can see here, CBDVA correlated with the CBNA isomer, and here, 2 peaks, CBNA correlated with the delta-9 THC isomer. However, both pairs didn't have the same UV absorption spectrum. Now here's another example. CBDV correlated with a delta-9 THC isomer. In this case, though, both of them have similar UV absorption spectrum. Now this peak was supposed to be a CBNA, but actually it showed up as a delta-9 THC isomer. This pair, though, had a different UV absorption spectrum. Now, LC-ESI-MS achieved comparable results as LC-DAD-ESI TOF-MS. However, LC-ESI-MS was less suited for compliance testing of hemp and hemp-derived products. We give a few reasons over here. It was more complicated with the procedures, and it has to use expensive, stable, isotopic labeled internal standards, and you cannot use all of them. Okay, we only used 6. Most importantly, MRM could not definitely distinguish structure isomers of delta-9 THC, and we had 7 of them. So their baseline separation was essential for their accurate quantification. This was the same requirement by LCDAD, ESI TOF-MS. So why would choose this over this? Finally, let's discuss the essential aspects in practice to implement LCDAD with optional ESI-MS for compliance testing. First, it requires the best separation in the literature, and here is what we recommend. The column, Raptor Arc 18 column, we use 2 of them connected consecutively. Column dimension 150 by 2.1 millimeter, 2.1 micrometer particle size at the core shell. A solvent 0.5 millimole formic ammonium formate plus 0.02 formic acid at pH 3. B solvent acetonitrile, mobile phase 75%, acetonitrile, flow rate 0.4 milliliter per minute, and the injection volume 4 microliter. A few aspects. It was able to baseline separate the maximum number 18 major cannabinoids, including CBT, with the longest retention time in a relative short time, 90 minutes. It uses an isocratic elution, therefore avoiding baseline drifts, which could cause high LLQs and narrow linear calibration range. It also uses a low buffer concentration in the A solvent to achieve a low LLQ and a wide linear calibration range, 3 orders of magnitude. It is applicable for potency testing of hemp and hemp-derived products for non-psychotropic cannabinoids and delta-8 THC-based products. It is applicable for compliance testing of hemp and hemp-derived products for psychotropic cannabinoids-based products. We have additional data not included here. Now, what about other separations? Other separations is also appropriate, but with a more specific application. So this is what we recommend for a wide application. A simple solvent extraction procedure, including homogenization, ultrasonication, pulverization, centrifugation, filtration, dilution, is sufficient. Methanol has been proven to be the best extraction solvent. All extractions were based on the following principle. So a sample should be completely and homogeneously suspended into methanol for efficient recovery of cannabinoids. In case we are dealing with new unanalyzed samples, this is the principle to figure out the sample preparation procedures. Now, specifically for brittle samples such as plant materials and tablets, they were ground pulverized and then added into methanol under ultrasonication. Liquid samples such as drinks, water-soluble oils, and topical serum were ultrasonicated and then added into methanol under ultrasonication. Topical samples such as lip balm were melted, vortexed, and then added to methanol under ultrasonication. Topical samples such as body lotion and face cream were stirred, pulverized, and added to methanol under ultrasonication. Now, solid samples that are sugar-rich, those are special, including gummies, hard candy, and coffee. They were first frozen in the ground, then completely and homogeneously suspended into water under pulverization because sugars don't dissolve in methanol. And finally, the suspension was added into methanol under ultrasonication at a 595 volume ratio. Solid or semi-solid samples that are sticky itself or sticky under moisture, including snacks and cartridges, were first frozen and ground and then completely and homogeneously suspended into methanol under pulverization followed by ultrasonication. UV detection at multiple wavelengths is preferred for the best sensitivity and 0.02 microgram per milliliter LLOQ and improved selectivity. External calibration is recommended because it's simple. Calibration range 0.02 to 25 microgram per milliliter can be accessed. This calibration range can be converted for different kind of samples with percentage values, 0.1 to 125% for 20 microgram per milliliter hemp concentrate, 0.04 to 50% for 50 microgram per milliliter plant materials, 0.0008 to 1% for 2.5 milligram per milliliter for drinks, and 0.08 to 10% for 250 microgram per milliliter other hemp-derived products. Now, a single extraction has been proven to be exhaustive for recovery of cannabinoids prior to analysis. In our studies, ACBD or cannabinoids not naturally present in cannabis plants and commercially available as a reasonable price was spiked into each sample to track recovery of sample preparation in real time. In the literature, standard addition has been the popular practice. Optional ESI-MS should be used to confirm identity. We use the ESI top MS, indicated overall good specificity with no interference in the accurate quantification of delta-9 THC and dot-9 THCA. Only a few minor interferences in the quantification of other cannabinoids from the matrix at close to LLQ level. Other high-resolution mass spectrometers can be similarly employed. For low-resolution mass spectrometers, we recommend multiple ion monitoring using protonated molecular ions, except for CBGA, using as a dehydrated protonated molecular ion. All right, here are the conclusions. The key parameters to use LCDAD with optional ESI-MS for compliance testing of hemp and hemp-derived products have been elucidated. The essential aspects for best practice to implement LCDAD with optional ESI-MS for compliance testing of hemp and hemp-derived products have been discussed. Collaborative research on best practice to implement LCDAD with optional ESI-MS for compliance testing of hemp and hemp-derived products with crime labs have been proposed. Here is my contacting information. Some of the references used by this presentation are listed here. And acknowledgements. We appreciate the financial support by AIGA, NSF, WIU, and we appreciate the opportunity by Forensic Technology Center of Excellence. And a big thank you for your attending this webinar. Thank you very much.
All right, hello everyone and welcome. My name is Catherine Bollinger and I'm a forensic scientist here at RTI International, and I'll be your moderator for today and the rest of this cannabinoid series. So thank you, Dr. Song, for giving us this informative presentation today. If you have any questions, please put them in the Q&A pod and we will answer them during this time. All right, so one of our first questions for you, Dr. Song, is about your filtration process. And so the question is, during the filtration, were there any trial and error in which type of filter to use?
The filter.
And
Yeah.
the additional comment in that was that this person had worked in a cannabis testing lab, and they had to make sure that they had the different types of correct filters because they would see some retention of acidic cannabinoids for certain matrices.
Oh, okay. Well, we didn't find that to be what I say significant, affecting the quantification of the cannabinoids. So the it's not here. Okay. Okay, here. So the filtration, we use the 0.2 micrometer PTFE syringe filter. So this is the manufacturer we have. So, but I think probably for the filters, you have different kind of materials. So we didn't find that this one caused significant errors for, for our analysis. So that's why one of the things, with crime labs, so that's my proposition, we can have a collaboration and do further, validation on those procedures.
Okay. Thank you, Dr. Song, for answering that question for us. So our next one is, they were wondering if D8 was frequently detected in plant samples, and if so, at what levels?
Oh, D8, actually, it really depends on what kind of sample we are analyzing. So at first, actually, we analyzed a certain kind of D8 distillate, and the concentration is super high. So it can be 80%. However, those kind of products comes in all different, types. Like fortified plant materials, they can put a lot in there, they can put less in there. So our understanding is search. So in our early days, when we analyze those kind of samples, the concentration of delta 9 can be very high. But late analysis, we found out the concentration of delta 9 THC was lower. I guess they have a better, manufacture procedures, or the regulation is getting tighter or something. And in addition, delta 8 THC products, they always have delta 9 in there. They always have in there. So because the cutoff has to be there 0.3%, if the delta 8 product, you add a lot of them in there, then it's likely delta 9 will be increased. And I think the manufacturer right now, they are smart enough right now for certain product, they just add something in there, and so that the delta 9 THC is not exceeding 0.3%.
All right, thank you for that explanation, Dr. Song. Our next question is about when you're evaluating compounds for interference. The question is, were delta 6A THC,
Yes.
delta 10A THC, and delta 10 THC evaluated in your interference studies?
Well, for published, this is the separation we recommend. For the publication we listed here, we didn't do totally. However, we have continued the study, and so more data is going to be presented. And I can safely say that for this separation, the quantification of delta-9 THC, where is it? Over here, and delta-9 THCA is accurate. It's not going to be interfered by any other newly emerging psychotropic cannabinoids. I can tell you more in detail here. So the delta-9 11 THC is just ahead of this one. You have to use two columns, otherwise they are not baseline separated. And also the delta-10 2 isomers. In a lot of publications, they only indicate one that's not complete. 2 isomers, they usually are around here, very close to CBL. It will interfere CBL, but CBL were occasionally found in the sample. And if the separation was not good, it might interfere CBC. So that both the 2 isomers of delta-10 is about here. And for any separation I saw so far, okay, one of the delta-10 is going to correlate with delta-6A and 10A, about here. But it's not going to affect the quantification of delta-9 THC and delta-9 THCA. Some of them not affected either. And for the homologs, if you have a longer side chain, occur chain, they are usually in this area or beyond this area. CBT sometimes can correlate with THCP, but there's ways to deal with it. So yes, we did a lot of analysis with those, but the data is not ready to be published yet. But I can safely say they are not interfering for compliance testing. In other words, quantification of delta-9 THC and delta-9 THCA.
Okay, thank you. Thanks everyone for all of the questions that you've put in. There've been some great questions so far. So if you have any more, go ahead and put them in the Q&A and we'll answer it while we have Dr. Song with us. And thank you, Dr. Song, for giving us details in these questions and answers. Okay. Okay. Well, it looks like we don't have any more questions coming in. So if there are no further questions, we're going to go ahead and wrap up today's webinar. So feel free to contact our presenter if you have any further questions on his presentation and research or collaborations. So thank you so much for attending, and Dr. Song, thank you for being here with us today.
All right, thank you, everybody.
As you depart, we kindly ask that you take a minute or two to complete our post-event questionnaire. The link will be pasted in the chat and also appear as you exit the Zoom room. Your feedback helps us improve our outreach efforts and continue to provide the best experience possible. Thank you again for attending, and we hope everyone has a great day.